All right, welcome to Rad Onc Smart Review, the RadBio edition.
Glad to be here.
So today we're doing a deep dive into oncogenes, really fundamental players in the molecular side of cancer development.
Absolutely. We're going to explore what they actually are and critically how normal genes sort of make that transformation into these cancer-promoting entities.
Think of it like a tutoring session almost.
Exactly. We'll break down the basics, the foundations, and then really try to connect it all back to what you actually see in the clinic and, of course, what you need for your exams.
Okay, so let's start with the foundations then. We all know our cells have these genes, right? They meticulously control growth, division when cells mature.
The normal regulator.
Right. And these essential genes are called proto-oncogenes.
Yeah, and a good analogy, I think, is like the gas pedal in a car.
Okay. I like that. Go on.
Well, proto-oncogenes kind of control the speed, the movement of the cell cycle. They make proteins that, stimulate division when needed, inhibit differentiation sometimes, stop cell death. But it's all very tightly controlled.
Like using the gas pedal appropriately.
Precisely. But just like a gas pedal can get stuck,
Right.
these proto-oncogenes can malfunction.
And that's when trouble starts.
Right. When they get specific genetic changes or maybe their expression just goes haywire, they can become oncogenes.
Onco from the Greek for tumor you mentioned.
Yep. So oncogenes are basically mutated or abnormally expressed versions of those normal proto-oncogenes. They really contribute to cancer.
So sticking with the analogy, if the proto-oncogene is the regulated gas pedal.
Then the oncogene is like that pedal jammed right down to the floor. Constant acceleration.
Uncontrolled growth, uncontrolled division in the cell.
Exactly. And this is really key. The defining feature of an oncogene is what we call a gain of function.
Gain of function. Okay, what does that mean practically?
It means the altered gene product, the protein it makes, is maybe hyperactive or it's just produced way too much, or maybe it's made at completely the wrong time in the cell's life.
And the result is that unregulated proliferation and survival signal that really characterizes cancer.
Yes. And crucially, this gain of function usually acts dominantly. That's a really critical board concept.
Dominantly, meaning?
Mutation, a change in just one copy of the gene, just one allele can be enough. Enough to push that cell towards becoming cancerous.
Wow, okay. So just one hit, potentially. That's different from tumor suppressors, right? Where you usually need 2 hits.
Exactly. That dominant action is fundamental. A single critical mistake in that growth machinery can kickstart the whole runaway process.
Okay, so how does this happen? How does a normal, well-behaved proto-oncogene turn into this oncogene?
Well, there are 4 main ways, 4 main mechanisms we need to talk about.
All right, what are they?
We've got point mutations, gene amplification, chromosomal translocations, and retroviral integration.
Okay, 4 key mechanisms. Is there a way to remember those?
Yeah, you could use a mnemonic, maybe like PGCR.
PGCR.
Point mutation, gene amplification, chromosomal translocation, and retroviral integration. Might help lodge it in your memory for the boards.
PGCR.
Got it.
Okay, let's start with P-point mutations.
Right. So this is pretty much what it sounds like. A change in just a single nucleotide, a single letter in the gene's coding sequence.
Like a typo in the instruction manual.
Exactly, a tiny typo. But that single base change can lead to a different amino acid being put into the protein when it's made.
And if that happens at a critical spot in the protein,
then yeah, it can be a disaster. If it hits a key site, like the active site of an enzyme, or maybe a spot where other proteins bind, it can make that protein permanently active, or way more active than it's supposed to be.
and interestingly, the actual amount of the protein might not even change in this case.
Correct. It's the protein itself that's now fundamentally altered and misbehaving.
Okay, classic example time.
Definitely the RAS oncogenes. The RAS family, HRAS, KRAS, NRAS, you see these mutated all the time in many, many cancers.
And what do RAS proteins normally do?
They're small GT passes. Think of them as molecular switches. They bind GTP, this energy molecule.
Right, guanosine triphosphate.
Yeah, they bind GTP and hydrolyze it, break it down. And this switching is key for relaying signals from the cell surface down to the nucleus, basically telling the cell, okay, time to grow and divide.
So critical communication hubs.
Very critical. And the point mutations in RAS are often found in really specific spots.
It's like hotspots.
Exactly, hotspots, like codons 12, 13, or 61. A classic one is changing a glycine amino acid to a valine at codon 12.
Seems like such a small change.
It is, but it cripples the protein's off switch. Normally, RACE is active with GTP bound, then it hydrolyzes GTP to GDP to turn off, often helped by proteins called GAPs.
GTPase activating proteins.
Right. These mutations stop that GTP hydrolysis, or they make RAS ignore the GDAP.
So the result is?
A RAS protein that's just stuck on permanently signaling. Divide, divide, divide.
Okay, that makes sense. So that's point mutation. What's next on our PGCR list? G.
G is for gene amplification.
Amplification, meaning more copies.
Exactly. The cell ends up with multiple extra copies of a proto-oncogene in its DNA. Not just the normal 2, but maybe tens, even hundreds of copies.
Wow, and what's the effect of having all those extra copies?
It's pretty direct. More gene copies mean more messenger RNA gets made, and that leads to a huge overproduction of the protein.
So even if the protein itself is normal, just having way too much of it can cause problems.
Absolutely. You overwhelm the cell's normal control systems. Even a normal protein in massive excess can drive that oncogenic behavior.
Okay, examples here.
Oh, several really important ones. The MYC family, NMYC, LMYC, CMYC.
MYC, right. I've heard of NMYC amplification in neuroblastoma.
Yes, exactly. It's a known marker for aggressive neuroblastoma associated with a poor prognosis. And you see CMYC amplified in breast cancer, lung cancer, others too.
And what do MYC proteins do?
They're transcription factors. They control the expression of lots of other genes involved in cell growth and differentiation. So when you overexpress MYC, you push the cell hard towards proliferation and you actually block it from differentiating properly.
Okay, MYC. Any other big examples of amplification?
Another huge one is E13A, also called ERBB2.
HRR2. Very relevant in breast cancer.
Extremely relevant. Amplified in about 20–25% of breast cancers, also linked to more aggressive disease. HR2 encodes a receptor on the cell surface, a tyrosine kinase.
So more gene copies mean more receptors on the cell.
Right. Way more HR2 receptors studding the cell surface, leading to a much stronger signaling for growth and survival.
And this is clinically vital because?
Because we have targeted therapies. Drugs like Herceptin trastuzumab specifically target that overexpressed HR2 receptor.
Which really highlights how knowing the specific oncogene can guide treatment.
It's a cornerstone of targeted therapy, though, resistance can still develop, which is a challenge.
Right. Any others? EGFR.
Yes. EGFR, the epidermal growth factor receptor. That gene gets amplified in cancers like squamous cell carcinomas, glioblastomas. Again, more receptors mean overstimulated growth pathways.
And can you actually see this amplification, like, under a microscope?
Sometimes, yeah.
Okay.
Cytogenetically, you might see what are called homogeneously staining regions, or HSRs, on a chromosome, basically. Big bands where the gene is copied over and over. Or you might see double minutes, tiny extra bits of chromosome floating around carrying the amplified gene.
Fascinating. Okay, so we've done P and G. Next is C.
C is for chromosomal translocation.
Translocation, so moving pieces of chromosomes around.
Yeah. Bits break off from 2 different chromosomes and then rejoin, but swapped. Genetic material gets exchanged between non-homologous chromosomes.
Okay, and how does swapping chromosome parts activate a proto-oncogene? Seems there could be different ways.
There are 2 main ways this works. The first way is when the translocation leads to overexpression, sometimes called a promoter or enhancer swap.
Promoter swap, meaning the gene itself isn't broken, but it lands somewhere new.
Exactly. The proto-oncogene gets moved next to a really powerful promoter or enhancer sequence that's normally driving some other gene very strongly in that particular cell type.
Ah, so the perfectly normal proto-oncogene just gets put under new management, so to speak, that tells it to work overtime.
That's a great way to put it. The normal protein is produced, but way too much of it, or maybe in cells where it shouldn't be active, all because it's now controlled by this hijacked regulatory element.
The classic example here is?
MYC again, but this time in Burkitt's lymphoma.
Okay, so MYC can be activated by amplification or translocation.
Correct. In Burkitt's, the CMYC gene, normally on chromosome 8, gets moved. Usually it translocates to chromosome 14, right next to the genes for the immunoglobulin heavy chain, the IgH locus. We write that as T814.
And in B lymphocytes, which become Burkitt's lymphoma, those immunoglobulin genes are super active, right, making antibodies.
Incredibly active. So when CMYC lands next to that super strong IgH enhancer, it gets massively overexpressed. And that drives the uncontrolled B-cell growth typical of Burkitt's.
And sometimes it can move to other immunoglobulin loci, too.
Yeah, less commonly, it might translocate near the light chain genes on chromosome 2 or 22. Same principle, same result. MYC overexpression.
Okay, so that's one type of translocation overexpression via promoter swap. What's the second type?
The second way is by creating a fusion
The
gene.
fusion gene. Sounds like combining two things that shouldn't be combined.
Pretty much. This happens if the breaks on the two chromosomes occur within the coding regions of two different genes. When the pieces are swapped and rejoined, you literally create a new hybrid gene.
A mashup of two genes.
Right. And this fusion gene produces a novel fusion protein. This new protein often has completely altered activity, and frequently that new activity is oncogenic.
So here you're not just overexpressing a normal protein, you're creating a brand new, potentially faulty one.
Exactly. A brand new hyperactive protein often. The absolute classic example is BCR-ABL in chronic myelogenous leukemia, CML.
Ah, the Philadelphia chromosome.
That's the one. It's the result of a translocation between chromosome 9 and chromosome 22, E922. The ABL1 gene on chromosome 9, which is a proto-oncogene encoding a tyrosine kinase.
A signaling enzyme.
Yes, it fuses with a gene called BCR, the breakpoint cluster region gene on chromosome 22. This creates the BCR-ABL fusion gene.
And the protein.
The BCR-ABL fusion protein is a tyrosine kinase that's constitutively active. It's always on, constantly signaling.
Driving the myeloid cells in CML to proliferate uncontrollably.
Precisely. And again, this is a huge therapeutic target. The drug Gleevec, imatinib, was revolutionary because it specifically inhibits this BCR-ABL kinase.
Incredible. Any other fusion gene examples relevant for us?
Another important one, especially in solid tumors, is EWS-FLI1 in Ewing sarcoma.
Ewing sarcoma, right? A bone and soft tissue cancer.
Yeah. Usually caused by a translocation between chromosomes 11 and 22, T1122. This fuses the EWS gene with the FLI1 gene.
Creating what?
Creating an aberrant transcription factor. This fusion protein messes up gene expression patterns and drives the development of Ewing sarcoma.
Okay, translocation covered promoter swap or fusion gene. That leaves R in PGCR.
Right. R is for retroviral integration, sometimes called insertional mutagenesis.
Retroviruses.
Yeah.
These are RNA viruses that copy themselves into DNA, right?
Yes. They use reverse transcriptase to make a DNA copy, the provirus, which then gets inserted into the host cell's genome.
And how does that cause cancer? Is it carrying an oncogene?
Well, some retroviruses do carry their own oncogenes. We'll touch on that. But this mechanism, insertional mutagenesis, is about where the virus inserts its DNA.
Ah, so the location matters.
Critically, if the provirus happens to integrate near one of the cell's own proto-oncogenes.
It can mess things up just by being there.
Exactly. Two main ways. First, the viral DNA often contains very strong promoters or enhancers to drive its own genes. If it lands upstream of a proto-oncogene, those viral signals can accidentally drive overexpression of the cellular gene. That's called promoter insertion.
Okay, similar idea to the promoter swap and translocation, but driven by a virus this time.
Sort of, yes. The second way is that the insertion itself can physically disrupt the proto-oncogene, maybe landing right in the middle of its coding sequence, or disrupting its normal regulatory regions. This could potentially create an altered oncogenic protein.
Is this a major cause of human cancers?
It's probably less common as a primary driver in humans compared to point mutations, amplification, or translocations, but it's a really important concept for understanding gene dysregulation. It shows how external genetic elements can hijack our own cellular machinery.
Is there a classic example, maybe from animal models?
Yes, the avian leukosis virus, ALV, in chickens. It causes lymphomas, but it does so by integrating its provirus near cellular proto-oncogenes like c-Myc, causing them to be dysregulated.
Interesting. And you mentioned some viruses do carry their own oncogenes.
Right, those are called acutely transforming retroviruses. Viruses like Rau's sarcoma virus, which causes sarcomas in chickens, it actually picked up a chicken proto-oncogene, SRC, mutated it, and now carries it as a viral oncogene, or V-on-C-VSRC in this case.
So when that virus infects a cell, it directly delivers an active oncogene.
Exactly. Leads to very rapid cell transformation. These viruses are often defective, though. They need a helper virus to replicate. It's a different mechanism than the insertional mutagenesis we see with ALV.
Okay, that distinction is clear. So we've covered the four main mechanisms: PGCR, point mutation, gene amplification, chromosomal translocation, and retroviral integration.
We have, and understanding these is, well, it's just critical for oncology.
Let's maybe pivot to some clinical pearls now. Kind of synthesize the key takeaways.
Good idea. So pearl number one. Just remember that core concept oncogene activation is about a gain of function. The altered gene does something new or does too much of its normal job.
And linked to that, pearl number two.
Yeah.
This gain of function typically acts dominantly. Remember, a change in just one allele, one copy, can be enough to contribute to cancer.
Which, as you pointed out, is different from tumor suppressors needing that two-hit inactivation generally. It also means inheriting a germline activating mutation in a key proto-oncogene is usually not compatible with life, which is why we see fewer inherited cancer syndromes driven by oncogenes compared to tumor suppressors.
That makes sense. What's another pearl?
Pearl number 3. Identifying these specific oncogene activations, like finding a RAS mutation or HR2 amplification or the BCR-ABL fusion, is absolutely crucial in modern oncology.
For diagnosis, right, and classification.
Diagnosis, classification, predicting prognosis, how the cancer might behave, and critically, for guiding treatment.
Which leads to maybe pearl 4, targeted therapies.
Exactly. So many successful cancer drugs work by inhibiting these specific oncoproteins. Imatinib for BCR-ABL, trastuzumab for HER2, EGFR inhibitors, the list goes on, targeting the driver oncogene.
And is there radiation oncology specific pearl here?
Yeah, definitely. Pearl 5. Activated oncogenes can impact how a tumor responds to radiation. For instance, pathways involving RAS or RAF activation have been linked to radioresistance.
So knowing the oncogene status might eventually help tailor radiation treatments too.
That's the hope and an active area of research, potentially using targeted drugs as radiosensitizers, for example.
Great pearls. Okay, feeling ready for a quick board review blitz.
Let's test that recall. Hit me.
All right, question 1. A mutation in a single allele of a gene leads to a constitutively active protein kinase that promotes uncontrolled cell proliferation. This gene is best described as an A tumor suppressor gene, B DNA repair gene, C apoptotic gene, or D oncogene.
Okay, single allele, gain of function, constitutively active, promoting proliferation. Now that screams oncogene, so D.
Correct, D oncogene. The explanation is exactly that. Gain of function from a single allele leading to uncontrolled proliferation is the hallmark characteristic of oncogene.
Good.
Question 2. The Philadelphia chromosome, commonly found in chronic myelogenous leukemia, CML, results from which type of oncogene activation mechanism? A, point mutation in the ABL gene, B, amplification of the BCR gene, C, retroviral insertion near the ABL gene, or D, chromosomal translocation creating the BCR-ABL fusion gene.
Philadelphia chromosome, CML. That's the classic T922 translocation fusing BCR and ABL. So, gotta be D, chromosomal translocation creating the fusion gene.
Spot on. D is correct. The Philadelphia chromosome is the direct result of that T922 translocation, creating the constitutively active BCR-ABL kinase.
Okay, feeling good. Keep them coming.
Question 3. Amplification of which of the following oncogenes is frequently observed in neuroblastomas and is associated with poor prognosis?
Yeah.
A, KRAS, B, BCL2, C, NMYC, or D, EGFR?
Correct again. C, NMYC. NMYC amplification is a well-known, very important marker for aggressive neuroblastoma. Definitely one to remember for PEDS on clinic.
All right, last one. Last one for this round. Question 4. Which of the following viruses is not typically associated with causing cancer in humans by directly carrying a viral oncogene, or VONC, into the host cell? A, Rari sarcoma virus in chickens. B, Harvey-Marine sarcoma virus in mice. C, human T-cell leukemia virus, type 1, HTLV1. D, avian leukosis virus in chickens.
Okay, tricky wording. Not directly carrying a VONC. Rus sarcoma virus has VSRC. Harvey has VIRES. HTLV1 is complex, causes T-cell leukemia, but doesn't actually carry a classic VONC derived from a proto-oncogene. It uses regulatory proteins like TAX. Avian leukosis virus, ALV. That's the one we said uses insertional mutagenesis, right? It activates a cellular proto-oncogene. So ALV doesn't carry its own VONC. I'll go with D.
Excellent reasoning. D, avian leukosis virus is correct. ALV causes cancer via insertional mutagenesis, activating a host gene, not by bringing its own captured oncogene. HTLV1 is also technically correct in that it doesn't carry a transduced VONC like RSV, but its TAX protein acts oncogenically. ALV is the clearest example here of the insertional mutagenesis mechanism without carrying a VONC.
Good clarification on HTLV1.
So let's try to wrap this up. A quick summary of our deep dive on oncogenes.
Okay. The big picture, oncogenes are the drivers, the accelerators. They come from normal proto-oncogenes.
And they get activated through those gain-of-function changes we discussed.
Right. Point mutations, like in RAS. Gene amplification, like MYC or HER2. Chromosomal translocations, making fusion proteins like BCR-ABL or causing overexpression like MYC and Burkitt's. And sometimes retroviral integration. Remember PGCR.
And crucially, these changes usually act dominantly. One bad copy can be enough.
Exactly. Understanding these mechanisms and key examples, RAS, MYC, HR2, BCR, ABL, is just fundamental for your boards, for understanding cancer biology, and for appreciating how targeted therapies work.
It really paints a picture of that accelerator pedal being stuck down in cancer cells, driving that relentless growth.
That's the essence of it.
So next time, we'll look at the other side of the coin.
We'll tackle the tumor suppressor genes, the brakes on the system, and how losing them also contributes to cancer.
Looking forward to it. Keep reviewing these oncogene concepts, everyone. They really are foundational.
Absolutely. Keep up the great work with your studies.
Okay, that wraps up this deep dive. Remember, you can find more resources and practice questions at radonsmartlearn.com. And please subscribe to Rad Onc Smart Review for our next exploration. Thanks for tuning in.